Thank you so much for joining this corporate presentation for Rion as part of the Cell and Gene meeting on the Med. We appreciate your attendance of this virtual session. Rion was founded in 2017, and it's really based on 2 decades of research that we performed at Mayo Clinic in the field of regenerative medicine. And its main focus is really to develop novel regenerative exosomes for use in the treatment of unmet needs of patients. The flagship product that we have in Rion is called Purified Exosome Product, or PEP, and that'll be the main focus of this presentation. This is an off-the-shelf regenerative exosome therapy that Rion has developed and actually taken to clinical stage with securement of 3 active FDA INDs focused on wound healing and soft tissue healing in tandem with cardiovascular application. We also have a robust clinical developmental pipeline with significant preclinical data focused on women's health applications, orthopedics applications of exosome therapies, and most recently, the development of exosomal targeting for pulmonary diseases. So, main question that is asked is, why exosomes instead of stem cells? Well, based on our research in the cell therapy arena, in particular, focused on stem cells as regenerative modalities, one of the key findings we had is that stem cells actually secrete exosomes to perpetuate their regenerative effect. And so use of exosomes directly as key drivers of regeneration, we feel may provide a more efficient and more expeditious application of regenerative technologies. Exosomes are essentially small extracellular vesicles, and they are engineered by cells as a uniform product that's secreted by cells out of the endosome that carries a specific signal to drive molecular events in recipient cells. And so by identifying and purifying a particular exosome with high regenerative potency, we find that we're able to achieve more efficient regenerative endpoints. Key features that improve on the existing stem cell-based PRP or even MSC-derived exosome platforms that are currently available is that the product that we make in the form of PEP is fully off the shelf and is actually accessible at the point of use. We've demonstrated significant consistency in the product that's generated. And the way that we've scaled our biomanufacturing actually renders this product as a highly affordable technology, which overcomes a lot of the bottlenecks that we were seeing with cell-based and cell-derived technologies for widespread clinical applications. So PEP, we really feel, is a breakthrough in regenerative medicine in that it is the first example of a truly off-the-shelf, room temperature stable regenerative therapeutic. We derive these from purified platelets. We have an established manufacturing process that derives a exosome product that is uniformly potent and stable. We characterize these with hallmark exosomal markers. And actually, a key element in our biomanufacturing process is to ensure that the exosome lipid bilayer is not infringed upon. And that is what gives us the ability to have high cell entry efficiency, and as seen in this lower panel, actually a high efficiency in binding basement membrane proteins like collagen for biopotentiation effect. We have been able to validate a conformity and molecular consistency in this product and actually have significant wealth of data documenting the pharmacokinetics or in vivo biodistribution of this technology with different applications. So in terms of our biomanufacturing, we obviously have FDA compliant source material. Our manufacturing avoids any excipients. Currently, our exosomal yields are in excess of 10 to the 14 per manufactured lot. And our manufacturing process ensures safety, purity, and potency with over 4,000 clinical trial material already generated and released for use in different clinical trials. I believe that a game-changing feature in the PEP technology is the fact that this is room temperature stable, and we have validated shelf stability for out to 6 months with this technology. PEP mediates tissue repair through a polyvalent mechanism. It activates mitogenic events in local tissue progenitors. It is proangiogenic, donating a tyrosine kinase receptor to activate proliferation and organization of vascular progenitors. It donates several antioxidant proteins to avoid free radical generation in tissue, which is one of the reasons why tissue healing is difficult. And it immunomodulates by donating PD-L1 to polarize macrophages towards an M2 phenotype. Taking the biomanufacturing, the biopotency, and our in vivo efforts together, we've been able to secure several INDs for use of PEP in man. Our wound healing trial is active and recruiting right now for the treatment of skin graft donor sites. Myocardial infarction, a second target for Rion, has an active IND with the trial slated to begin recruitment in April of 2021. Fistulizing disease is an approved IND by the FDA, and we're in the planning stages for that trial. And we're planning several additional trials focused on women's health, orthopedics, pulmonary applications moving forward. This slide depicts the breadth of the preclinical work that has been performed in collaboration with colleagues at academic centers such as the Mayo Clinic. The triangles highlight the preclinical work that has led to clinical studies, and we continue to work in the realm of cardiovascular, derms, plastics, and surgery, women's health, orthopedics, and pulmonary to bring the exosome platform to fruition and ultimately translate these efforts to man. Some of the targeted efforts that have led to human applications include the finding that PEP promotes wound healing. We've been able to document that not only does PEP induce angiogenic events in vitro, it can also activate skin organoid formation in the Petri dish. And leveraging these findings, we've been able to document in large animal models, not only restoration of blood flow in the setting of ischemia, but also full thickness healing of skin in ischemic wounds. For cardiovascular application, we've been able to show that through intracoronary delivery, PEP can be targeted only to the area of injury following myocardial infarction. And indeed, we have functional data documenting that in contrast to placebo-treated animals where a scar ensues following myocardial infarction, following PEP treatment, we can achieve cardioprotection in the tissue area at risk, ultimately resulting in functional improvement that's documented both on MRI and hemodynamic catheterization studies. We have also a significant effort in women's health applications, applying PEP to both models of stress urinary incontinence in tandem with vaginal mesh erosion. In both of these areas, what we note is a significant improvement in the soft tissue performance in stress urinary incontinence, we show that in contrast to the vehicle, PEP treatment fully restores the functional integrity of the urethral sphincter in the porcine model. And in vaginal mesh erosion, we note that application of PEP to non-healing meshes achieves full thickness restoration of the vaginal mucosa. All of these efforts have now been under peer review and have been published in the literature. We also have a significant academic effort in orthopedics looking at PEP for both tendon healing as well as peripheral nerve graft healing. And in both of these areas, we have numerous publications now in the literature documenting that PEP drives intrinsic healing of tendons in both rodent and rabbit models of tendon injury. And we show that this tendon healing heals the tendon without scar formation, achieving high strength despite a lower profile of the tendon, which is consistent with normally healed tendon. So I think in summary, what we can highlight is that PEP is one product that has multiple therapeutic applications. We can deliver this topically or subcutaneously, intravenously or intraarterially. It readily dissolves in any aqueous solution, and as such can biopotentiate hydrogels for controlled release, or can be used at the point of use, such as in myocardial infarction, for acute illnesses that to date have not been able to be treated with regenerative technologies. The phase 1 clinical study for skin graft donor sites is now underway and is actively recruiting patients by invitation only. This will treat donor sites with PEP or PEP plus fibrin glue, and that trial will be carried out at the Mayo Clinic. The second trial that is slated to begin in April is a myocardial infarction trial, where patients presenting with acute myocardial infarction and undergoing PCI or stent placement in the setting of a heart attack will be recruited in the emergency room for treatment with exosomes. And after stent deployment, which restores blood flow to the injured tissue, the exosomes will immediately be delivered with an effort to assess their safety and also their efficacy in restoring blood perfusion and protecting the heart against further injury. We are based in Rochester, Minnesota, across the street from Mayo Clinic, and are building an R&D and manufacturing facility that'll be open in the middle of this year. And we have a very robust management team who, in tandem with my co-founder, Andre Terzic, are now really leading us to be able to establish this platform exosomal technology for use in man. I thank you very much for your attention. This is our contact information if you would like further information.